Good morning. Today we are going to discuss the topic about the overview of a fermentation process. So before entering into the topic, let me know how many of you came to this topic fermentation. Yes? Okay. So now we will see what are the topic which we will discuss in the fermentation. So we will discuss about the definition. And the next we will see about the types involved in the fermentation process, and the third one we will see about the process that occurs in fermentation, and the fourth one we will see the range of fermentation, and we will see one example of fermented food. Okay. So now we will enter into the definition. So coming to the definition, the term fermentation is derived from the Latin word fervere. So fervere, which means boil. Okay. So I will tell what is the meaning of boil. So in the ancient days, people used the pots like mud pots for the fermentation process, and they have used some of the malted grains or the fruits by the action of yeast. So what will happen is, when we are adding the malted grains or the fruits onto the fermenter. So here I have mentioned the fermenter as the mud pot. When we are introducing the yeast, so this yeast will utilize the sugars, that is the carbohydrate that is being present in the malted grains or the fruit, and that way it will convert this into either acid or alcohol. So that we can call it as fermentation. Okay, so due to the appearance of the boiling, so the term fermentation was originated. Now we will see the types of fermentation. So coming to the types, generally the fermentations are being categorized into two forms. One is lactic acid fermentation, and the other one is alcoholic fermentation. So what is the difference between the lactic acid fermentation and the alcoholic fermentation is, this lactic acid fermentation will carry out in the anaerobic process, whereas the alcoholic fermentation will be carried in aerobic condition. Students, what is the difference between the aerobic and anaerobic conditions? Yes. Okay. So depends upon the oxygen requirement, the process may be called as anaerobic or aerobic. So in case of anaerobic process, the process does not require oxygen. And in the case of aerobic condition, the process will require oxygen for the fermentation. Okay, so that is the main difference between anaerobic and aerobic condition. So as I told that in case of lactic acid fermentation we will use anaerobic process, and in the alcoholic we will be using the aerobic condition. So what will happen in case of lactic acid fermentation is, so here the glucose molecules, that is glucose, which is an simple sugar, which will be converted into lactic acid by the action of microorganism and the process that is called as glycolysis. So glycolysis is a metabolic pathway in which the glucose is being converted into an pyruvate, and thereby the formation of lactic acid will be produced, and this lactic acid will be generated as a byproduct. So that's why the lactic acid fermentation is called as lactic acid fermentation. And the example of lactic acid fermentation is, we can consider the best example as yogurt. And what is the microorganism involved in the yogurt is, so lactobacillus will be involved. Okay, so I think you are clear about the lactic acid fermentation. Now we will move on to the alcoholic fermentation. So as I told, the alcoholic fermentation will be carried out in aerobic process. And here what will be the product formed is, the product formed will be ethanol, and the microorganism that is being involved in the ethanol is yeast. And one such example of the yeast is Saccharomyces cerevisiae. Hope you came to know about the types of fermentation. Now we will see about the process in which how the fermentation is being carried out. Coming to the process, so generally the glucose molecule, that is glucose is a sugar, simple sugar, and which will be converted into either pyruvate or lactic acid. Okay. And if it is the product formed as lactic acid, then the microorganism which is utilized is lactobacillus. And if it is of pyruvate or ethanol, the organism is yeast. So in the yeast, the main category is Saccharomyces cerevisiae. Okay, so in that way the glucose is being converted into a different products under the process, under the metabolic pathway, that is glycolysis. So normally it is a metabolic process in which the substrate is being converted into a product. Okay. Next we will see about the different ranges of fermentation process. The ranges of fermentation, for example, the fermentation may be varied into different categories. It ranges from aerobic to anaerobic, then it ranges from batch to continuous and so on. Okay. As well as depends upon the various factors such as the type of microorganism which is being involved, and depends upon the substrate that is being utilized for the fermentation, and depends upon the product. The range of fermentation may vary, and we can categorize it into based on the oxygen requirements, and based on the modes of operation. So these are the different ranges, and let me tell each step in a single slide. Okay, so for example, I told the ranges of the fermentation can be applied to various of variety of byproduct, variety of products. First one, it can be utilized for the production of microbial cells or the biomass. It can be used for the production of microbial metabolites. It can be applicable for the production of microbial enzymes. Also, it can be used for the production of recombinant products. And for the transformation. So for those purpose we can apply this fermentation for production of variety of byproduct. So we will start with the microbial cells or the biomass. So initially we are in need of the microorganism in order to produce the cells, and what kind of product will comes under this category is one is single cell protein. So in short form we can call it as SCP. So it is a protein, single cell protein. And in case of microbial enzymes, there are various enzymes that fermentation product can produce, such as we can consider the example as protease and so on. And coming to the microbial products, when a microbe is being grown in a particular nutrient media, the microbes will start to produce various metabolites, and these metabolites are being categorized into two forms. One is primary metabolite and the other one is secondary metabolites. And this primary metabolite is very much essential for the growth of the microorganism, and some of the examples are amino acids, nucleotides, then proteins. Whereas the secondary metabolites are the antibiotics, and which is not essential for the growth of the microorganism. And the next example, next product is recombinant product. So here with the help of RNA technology or the genetic engineering, we can produce the products such as insulin. And the last one is transformation. So here what will happen is substrate is being converted into a product. So during that some of the metabolites will be produced. And the best example is steroids. So there are 5 different ranges by which fermentation can be varied. So one is microbial cells or the biomass, and the second one microbial enzymes, and the third one is microbial metabolites, and the fourth one is the recombinant product, and the final one is the transformation. Okay, and I have explained that the ranges of fermentation will vary from aerobic to anaerobic, then batch to continuous, right? So coming to the aerobic, so based on the oxygen requirement, it is being categorized into aerobic and anaerobic. Okay, so in case of aerobic it needs oxygen, and in case of anaerobic it does not need oxygen. And based on the mode of operation, it is classified into 3 forms. One is batch fermentation, and the second one is fed batch fermentation, and the third one is continuous fermentation. So what is the difference between the batch, fed batch and continuous is, in case of batch fermentation, to carry out the fermentation we have to prepare the media, and we are in need of a particular substrate. So we have to prepare, we have to make the essential media, and thereby we have to inoculate the microorganism, and after the reaction we will collect the product. In case of fed batch, So we have to prepare the media, and here the intermittent substrate addition is being carried out in case of wet batch. Whereas in the continuous, we will add the substrate and we will collect the product simultaneously, and the utilized substrates are being cells or the products are being collected by simultaneously. So that is the main difference between batch, wet batch and continuous. Okay. So one best example for the fermentation process I will explain. So the preparation of curd. So this curd is a best example of the fermented product. Okay, and there are several steps involved in the curd preparation. First one is we are in need of the preparation of the milk. And the second step is we have to inoculate the starter culture. And the third step is fermentation. And the fourth one is curd formation. And the fifth one is storage. Okay. So I will explain each step. So in case of preparation of milk, so we have to collect the milk, then we have to boil the milk under particular temperature. Why we have to boil is in order to kill the unwanted bacteria that is being present in the milk, and we have to maintain the milk at a temperature condition of 37 degrees Celsius to 40 degrees Celsius, and this temperature which is called as lukewarm temperature. Okay, so now the milk is being ready, and the next step is we have to inoculate the starter culture. So how we are going to introduce the starter culture? So already we have the curd, then in that curd some of the live bacteria is being present and which is being introduced onto the prepared milk. Okay, so here the live bacteria is being introduced. So some of the bacteria is lactobacillus, and the third one is we have to make it into a fermentation. So during the fermentation we have to maintain the lukewarm condition, then only the milk will be converted into a curd. So in the milk there will be a sugar that is lactose. So this lactose is a milk sugar, and this will convert into lactic acid with the help of a bacteria, and this lactic acid will make some changes in the pH of the milk, and that way which will coagulate the protein that is being present in the milk, that is casein. And the last step is curd formation. So the formation of curd will require some 4 to 6 hours. Okay, and that way the thick consistency will be formed. And the last step is storage. Why we have to store is in order to control the growth of microorganism, and here comes the example of the curd formation. Okay, so here you can see we have prepared the curd with the help of the preparation of the milk, then we have introduced the starter culture into the milk, and that way the fermentation process is carried out, and finally the curd is formed. And in order to prevent the growth of microorganism, we have to store in refrigerator condition. So this fermentation process is being applicable in various food industries as well as pharmaceutical industry. Thank you.